Really the highways that these cells we're going to talk about now use to move from place to place, the lymph nodes and the lymph vessels. That's a very extreme over exaggeration, but you can think of it that way. Now we're going to look at the cells that protect us, the cells and things that protect us against infections. So the lymphatic system defends the body against infections by pathogens. The definition of a pathogen is something that causes a disease. They typically are bacteria, viruses. They can be more complex, like protozoa, spores of multicellular organisms like fungi. Uh, um, that would include parasitic worms, for example, in the protozoa. Presence and multiplication of a pathogen in the body, if unchecked, can cause an infection, and there are a number of mechanisms that help to prevent them from ever getting into the body, and then there are others that help defend us if they do get in. So we have some innate or non-specific defenses, which would include the general defenses, and those protect against many different pathogens. They're not quite as specific as our adaptive or specific defenses. The adaptive defenses are known as immunity. The two defense systems kind of get interchanged a lot, especially in these days with lots of public discussions around immunity and defense against disease. But in general, the adaptive defenses are much more specific. They are very precise. They specifically target antigens. Not only that, the molecules they produce specifically to target pieces of antigens known as antigen determinants. Those are lymphocytes that recognize foreign molecules, either B lymphocytes that produce antibodies or T lymphocytes that produce T cells that have to actually come in contact with an antigen to do their job. So non-specific defenses would include a species resistance. So some species are resistant to diseases that other species are susceptible to. We have mechanical barriers. Skin is a very good one. Mucous membranes have to allow for some exchange, but they're still very good at keeping out infectious agents, washing them away before they can attach to tissues. Inflammation is a process by which we use chemicals to non-specifically protect us against pathogens. There are a number of chemical barriers that chemicals produce that will help fight off pathogens. Natural killer cells are kind of an in-between cell, in between specific and non-specific. They kill things, kill cells that do not have our own markers on them. Phagocytosis, the eating of pathogens by neutrophils, monocytes, macrophages, and even fever helps to protect us from disease. So there are certain species that do not have the appropriate temperature or chemical environment for a particular pathogen to survive and proliferate. So certain species are resistant to diseases that affect other species. They're permissive, I think, is a good way to think of that. There are certain pathogens that are permissive in one species but not permissive in another. Again, those mechanical barriers are very important. The skin and mucous membranes are mechanical barriers. They prevent the entrance of pathogens. They are considered the first line of defense. Anything that gets past skin and mucous membranes has to deal with our defensive system, and those would all be considered the second line of defense. As epidermis sloughs off, bacteria that attach to that will go with the sloughing off epidermis. Ciliated epithelium in the respiratory tract helps to sweep away pathogens. Hair helps to trap pathogen, hair in our nose and our ears in particular. And tears and saliva and urine all wash away microorganisms. In addition, several of those contain enzymes that are antimicrobial in nature. Chemical barriers include enzymes in body fluids that provide chemical barriers to pathogens. Pepsin in gastric juice, lysozyme in tears destroy microorganisms. There are chemicals called interferons that are involved in blocking viral replication. They They may work against growth of tumors and stimulate phagocytosis. Defensins are peptides produced by neutrophils and other granulocytes that cripple microbes by poking holes in their membrane or walls. Collectins are proteins that protect against many bacteria, yeast, and some viruses. I am not particularly familiar with collectins. I'll have to look into that further. Complement is a group of proteins in the plasma and other body fluids that stimulate inflammation, attract phagocytes, and enhance phagocytosis. Natural killer cells. NK cells are a small population of lymphocytes. They are very different from the B and T lymphocytes, though, that provide adaptive defenses. Natural killer cells provide protection because they look for cells that don't have our own markers on them, whereas B cells and T cells are truly going to look for antigens like we will be talking about for most of the day today. Natural killer cells, in addition to killing directly, are also very good at initiating an inflammatory process, which is an important part of getting an immune process going. Inflammation, there's some produces redness, swelling, heat, and pain. It's a process that helps to inhibit the spread of infection by walling off the site and limiting the use of that tissue. So blood vessels dilate, capillary permeability increases, fluid leaks into the tissue spaces. That leads to the redness, swollen, warm, and painful symptoms of inflammation. White blood cells are then drawn to the area. That can lead to pus formation. The tissue fluids that contain clotting factors seep into the area, and those clots form in kind of a spherical shape around the area. Fibroblasts will come in, phagocytes become active, and cells will begin to divide to replace the injured cells. So it's an important part of inducing an immune response and setting up the site for healing. So phagocytosis removes foreign particles from the lymph. Phagocytes in the blood vessels and the tissues of the spleen, liver, and bone marrow would get their particles from the blood. Most active phagocytic cells are the neutrophils and the monocytes. Chemicals from those damaged tissues will attract those phagocytic cells using a process called positive chemotaxis. Monocytes that leave the blood in the tissues can become macrophages, and they can either roam around freely in those tissues or become fixed in place in those tissues. And the mononuclear phagocytic system, which would be the reticuloendothelium, consists of monocytes and macrophages of the body. Fever. Fever begins whenever you have a viral or bacterial infection that stimulates lymphocytes to proliferate, and some of those cells will produce a substance called interleukin-1. And interleukin-1 is a type of chemical known as a pyrogen. It's an endogenous pyrogen. It resets the body temperature to a higher level. And that fever is important because it causes the liver and the spleen to sequester the iron they have. And iron turns out to be a limiting nutrient for the proliferation of bacteria and some viruses. In addition to that, the higher body temperature increases some of the enzymatic activity of our cells. The specific defenses are a third line of defense. It takes a while to get these going. Once they're going and running, they are very good at what they do, but it does take a while to get them going. Their response is based on the ability to distinguish parts of the body from things that are not parts of the body. So self versus non-self. So antigens are non-self molecules that can cause an immune response. The adaptive immune responses are carried out by lymphocytes, and it can be in one of two types of defenses. The cellular immune defenses involve the T cells, while the humoral immune response involves the B cells. The cellular response requires a T cell to come in direct contact with a pathogen, whereas the humoral response produces antibodies, and those antibodies are sent to all the body fluids most body fluids, to protect us against pathogens. So a pathogen begins to enter the body, and it would first encounter our first line of defense, the mechanical barriers. If it gets through those, it would, and we haven't seen it before, it would make its way, have to fight its way through this second line of defense. So those chemical barriers, natural killer cells, inflammation, phagocytosis, fever. If we have seen it before, then as soon as it gets in the body, our third line of defense helps to protect us. But usually if it's a new pathogen, that third line of defense is going to take a week or two to become active and create a cellular immune response and a humoral immune response. So antigens are receptors on the surface of lymphocytes that allow them to recognize non-self antigens. Those antigens could be proteins. Those are very good antigens. Polysaccharides in some cases can be antigens. Glycoproteins, glycolipids. The most effective antigens tend to be large, complex molecules, and there's variability in them. They are different from individual to individual. There are some antigens known as haptens, which are not antigenic by themselves, but when they combine with a larger molecule, they become antigenic and induce a immune response. So lymphocytes are produced throughout life, starting during fetal development. Eventually, the red bone marrow releases unspecialized lymphocyte precursors into the blood. Half of those settle in the thymus. They specialize and then are released. The other half of cells remain within the red bone marrow, and they are educated and released as B cells. So T cells, or T lymphocytes, T cells as I refer to them, are thymus-derived lymphocytes. Actually, they're made in the bone marrow, and they mature in the thymus. So they become effective in the thymus, and after they leave the thymus, they make up 70 to 80% of circulating lymphocytes. Some of them go to lymphatic organs, such as the lymph nodes, thoracic duct, white pulp of the spleen. The B lymphocytes, B cells, B does not stand for bone marrow. It stands for the bursa of Fabricius, and that is an organ that is found in birds, and in particular in chickens, where these lymphocytes were first isolated. So after they're released from the bone marrow as mature cells, they make up about 20 to 30% of the lymphocytes in blood. They are found in lymph nodes, spleen, bone marrow, and the intestinal lining. And so I won't walk you through 16.17. You should be able to follow that on your own. T cells and B cells, they both originate in red bone marrow. The thymus T cells differentiate in the thymus, B cells in the red bone marrow. 70% for T cells, 30% for B cells in the blood. And T cells provide cellular immune responses, but they're also very important for getting humoral immune responses going. B cells provide the humoral immune response. They produce the antibodies. So a lymphocyte has to be activated before it can respond to antigen, and that requires the use of an antigen-presenting cell, an APC, and those tend to be macrophages, B cells, or some other types. Antigen-presenting cells phagocytize antigens, digest it, display its fragments on its own cell membrane, bound to a protein that's part of the major histocompatibility complex, or MHC, sometimes referred to as human leukocyte antigens. When a specific T cell encounters an antigen-presenting cell that displays its target antigens, it can become activated and secrete polypeptides, which are called cytokines, and those are hormone-like substances or that cause other cells to do things. T cells come in three major types: helper T cells that activate other T cells, help B cells produce antibodies, generally help with an immune response. And cytotoxic T cells will attack directly virally infected or any cell that bears its target antigen on it. And either one of those can become memory T cells, which provide protection against future encounters with the same antigen. Some of those cytokines are colony stimulating factors, interferons, interleukins, or TNF, tumor necrosis factor. They all have different activities. They're all small chemicals produced by cells acting on cells around them. So they're not really like hormones. They don't tend to have systemic effects, but they do have localized effects. B cells are activated when an antigen that fits the shape of their receptor binds to them. Their receptor is an antibody, an IgM antibody on the surface of a B cell. Once that happens, and if there is help from T cells, the B cells will begin to produce more of their antibody, and their antibody will become more specific as it matures. Some of the B cells will become memory cells to protect against future infections. Many of them will differentiate into plasma cells that will pump out large amounts of the proteins we call antibodies. Those antibodies are carried by the blood or some other body fluid, so this is a humoral response, also known as an antibody-mediated response. Both B cells and T cells require proper activation. Helper T cells can help with that activation, and B cells can help activate helper T cells. They all lead to proliferation, producing memory cells and antibody-producing cells. In the case of B cells, in the case of T cells, we have memory cells, and then we have cells that actively engage infected cells. I'm not going to walk you through 16.6, but I think it's important that you do go through this table and as part of your reading. So antibodies themselves are globular proteins. In fact, they make up the gamma globulin fraction of plasma proteins. They are also called immunoglobulins. They have a Y shape to most of them, 4 amino acids, which are composed of 2 heavy and 2 light chains. Each type of antibody has a unique sequence and conformation that makes it very specific for its antigen or for a determinant on its antigen. There are 5 major classes of antibodies. They are IgG, IgA, IgM, IgD, and IgE. IgG is found when secreted, found in the plasma and tissue fluids. IgA is found in secretions, also are sent through exocrine glands. IgM is found mostly in plasma of blood. IgD is involved in the development of B cells, to the best of my knowledge, unless it's changed since I have left actively researching in the field. It's not something that's used for fighting pathogens, but it's used during the developmental process of B cells. IgE is also a secretory antibody. IgE gets a bad name because it tends to promote inflammation and especially allergic responses. Antibodies can react to antigens in 3 basic ways: a direct attack, activating complement, or stimulate local inflammatory to help prevent the spread of infection. Immunotherapy makes use of immune system components to fight diseases, either humoral with antibodies or cellular immune by the use of cytokines. Monoclonal antibodies are a specific single type of antibody produced from a single progenitor cell, producing one antibody. And to do that, you take an antibody-producing cell and fuse it with a cancer cell. Those are used in research, agriculture, many medical tests. A pregnancy test is based on monoclonal antibodies. The antibody test for COVID. The rapid test that you may have gotten or heard of from Walgreens is an antibody, is a monoclonal antibody test. Cytokines produced in large quantities because of recombinant DNA. They can be used to help treat leukemias, multiple sclerosis, many other disorders. So a primary immune response is produced by the first encounter with the antibody. First antibodies don't appear for 5 to 10 days. They remain for several weeks, and some memory B cells are generated. In a secondary immune response, the next exposure to an antigen gives you a stronger and faster response. You get higher concentrations of antibodies, and they appear within 1 to 2 days. Those antibodies remain for months or years, and memory B cells can live for many, many years. This is the idea behind the 2-dose regime of the Pfizer and Moderna vaccinations. You give a dose, wait 3 or 4 weeks, give a second dose, gives you a higher response and a more protective response. So immunity can be classified. Naturally acquired would be those obtained by a natural process, getting and recovering from a disease given from a mother to an infant. Artificially acquired would be obtained by an injection. Passive immunity is temporary immunity that you get when you are given antibodies but no antigen exposure, so you don't get your own immune response. And active immunity is a permanent, hopefully, immunity obtained through contact with the antigen, the production of memory T cells and B cells. There are some issues we can have with immunity, including hypersensitivity, which is an exaggerated immune response to something that's not really harmful. Person first has to be pre-sensitized. You have to have that first exposure, and they can affect almost anyone. Some can affect only people with inherited ability to have the immune response. So again, there's a connection between genetics, but it's not a genetic disease as such. So a type 1 hypersensitivity reaction is an immediate reaction. That's also called an allergy. People produce IgE antibodies against a specific allergen, and those occur quickly, minutes after contact with the allergen. The release of histamine, and histamine induces things like hives, hay fever, asthma, eczema, gastric disturbances, and this can be life-threatening. And in some cases, we can get what is known as anaphylactic shock. Bee stings can do that. You should carry an EpiPen if you are allergic to bees. Type 2 hypersensitivities are antibody-dependent cytotoxic reactions. Here, an antigen binds to a specific cell, causes phagocytosis, complement lysis of the antigen, and a transfusion reaction is a type 2 reaction. A type 3 hypersensitivity is an immune complex reaction. So the antigen-antibody complex form, and then they get deposited in tissues and damage to the tissues by phagocytosis and complement binding. Those antibody complexes aren't easily cleared from the body. Rheumatoid arthritis is typically a type 3 hypersensitivity. A type 4 is a delayed type hypersensitivity. They can also affect anyone. And that results from exposure to skin allergens. The allergen activates T cells within the skin. They release chemicals that cause the eruptions and inflammations, and they typically take about 48 hours to first appear. There's an immediate. So we also have to talk about transplantation and tissue rejection because the immune system mediates that as well. Tissues and organs that can be transplanted would include the cornea, kidney, liver, pancreas, heart, bone marrow, skin, bones, lungs. When they are recognized as foreign, there is a tissue rejection reaction. So the immune system of the patient tries to destroy the transplanted tissue, very similar to the cellular immune response against antigen. For that reason, it's important to match the antigens between the donor and the recipient, and some of the important antigens include that MHC group of antigens, the major histocompatibility complex antigens, and that's really what the name histocompatibility refers to. The stronger or more different they are, the stronger and faster the reaction will be. We can use immunosuppressive drugs to prevent rejections. Those immunosuppressive drugs, except in the case of bone marrow, have to be given for the rest of the life of the patient who receives the transplant. So we can have isografts, autografts, allografts, or xenografts. Isografts are between identical twins. Autografts are taking tissue from one part of your body and moving it to another. In both of those cases, do not have to worry much about, at all, honestly, of tissue rejection, because the tissue is basically self-tissue. An allograft is a donor of the same species, a kidney transplant from a living related donor, or even a kidney transplant from a non-related donor. Those work very well, but those patients have to take immunosuppressive drugs for the rest of their lives. Xenografts are from a different species, and generally those don't work very well, but we can replace heart valves from a pig in humans, and they have to be watched closely for tissue rejection. Autoimmunity is when your immune system attacks itself, its body, fails to distinguish self and non-self, produces antibodies called autoantibodies or cytotoxic T cells that attack tissues and organs. There are various autoimmune disorders that affect types of cells. We've talked about some with each system we've talked about. We'll talk about more with each system we come to. There are lots of theories how autoimmune disorders happen, but we do not have any single causes established. I won't walk you through 1612. Those are very common autoimmune disorders, some of which you may have heard before, some of which you may not have. The immune system function declines early in life as the thymus gland shrinks. There's a higher risk of infection and cancers later in life. T cell numbers decrease slightly. B cell numbers don't change. Activity levels change, both types of lymphocytes. Antibody response to antigens becomes slower. IgA and IgG antibodies tend to increase, while IgM and IgE antibodies tend to decrease. Elderly may not be candidates for certain medical treatments that would suppress immunity, and that would include chemotherapeutics. Human immunodeficiency virus is a virus that breaks down the immune system. It can stay silent for years and then progress to AIDS, or acquired immune deficiency syndrome, which would lead to opportunistic infections. HIV attacks macrophages, and from there it goes to helper T cells. It really gets rid of the CD4 positive helper T cells. Without helper T cells, the B cells don't do what they are supposed to do as effectively. HIV variants can also affect cytotoxic T cells, but cytotoxic T cells also require helper T cells. Eventually, a person would die from the loss of immune response against pathogens or cancer. HIV is transmitted through sexual contact, contaminated needles, birth or milk from an infected mother, or receiving blood or tissues from an infected donor.